One way to study the past is through the study of ancient DNA, which can not only tell us about the origins of diseases, but also about the people who had them. Together with historical and archaeological evidence, it contributes to our picture of the social, economic, and political conditions of the time. By studying the evolution of pathogens, we can better understand what kind of medications and vaccines may be needed to keep the human population healthy in the future. The study of ancient DNA has revolutionized our understanding of evolution, genetic diversity, and human history. But why is studying ancient disease so important? In the past 40 years, the study of ancient DNA evolved from a laborious and limited way to study biological history to an important research method for understanding the past. Scientists have found the study of ancient DNA to be a vital asset in the search for answers about the origins of pathogens and how human populations were impacted by disease. We can also see how pathogens have been one of the greatest evolutionary pressures on the genes we carry today. In the early 20th century, just as the world saw the beginning of the end of the Great War, the Spanish flu outbreak began. This pandemic swept the world in under 4 months and eventually took the lives of at least 20 to 50 million people, more than any other known disease previously. This flu pandemic is now known to have been caused by an H1N1 virus that originated from strains that infected birds. Scientists were able to recover the sequence of the 8 genes that make up the virus and study which parts of the genes were directly related to bird strains. Researchers have been using DNA samples from this pandemic and other pandemics to try and understand the origins of these pathogens and how they evolved. But why was the 1918 flu pandemic so devastating? The hemagglutinin and neuraminidase genes that encode proteins important for virus entry and exit do not indicate any changes that would make the pathogen have such a high mortality rate. Even after recovering the full genome from the 1918 flu virus, researchers still have many questions about what can cause a pathogen to become pandemic. And by further studying these diseases, we can create a better understanding of the evolutionary path of pathogens causing epidemics, such as the Spanish flu and COVID-19, to prevent future pandemics. In this regard, let's delve deeper into another ancient disease, the Black Death. It caused over 75 million deaths. And by studying ancient DNA, scientists have confirmed that the bacteria, Yersinia pestis, caused not only the Black Death, but the earlier Justinian plague as well. Also, ancient DNA of individuals from the late Neolithic and Bronze Ages revealed that Yersinia pestis was present even earlier and changed over time in ways that facilitated its spread in a flea vector. The study of ancient DNA also helps us recover pathogen genomes from challenging modern clinical samples. One example of this is ancient DNA analyses of Mycobacterium lepra, the causative agent of Hansen's disease or leprosy. Though ancient DNA data revealed that Hansen's disease began to affect humans about 4,000 years ago, today there are approximately 200,000 new cases each year. By having an understanding of ancient or historic genomic diversity, we can also track drug-resistant mutations through time and space and better address the unique needs of different regions affected by leprosy or other pathogens. Ancient DNA research has also provided valuable insight into pathogen jumps. For example, it revealed that ancient American tuberculosis spilled over into humans from contact with seals, an important insight into its evolutionary history and possible future. The study of DNA from ancient pathogens and their human hosts can assist in understanding why certain populations are more vulnerable to a disease than others. Ultimately, the examination of ancient DNA is a crucial tool in comprehending the history of diseases, such as the Black Death, leprosy, influenza, and tuberculosis, and developing more effective strategies for combating them in the future.